Okay, guys, let's get started. We've got a lot to do. Okay, so business. Recitation starts on Monday. That would be today. Did I not fix this slide? Sorry, this was like the same business slide from the other day. Anyways, I meant to alter it. But recitation starts today, downstairs in 217. When you come in, just hang out for a minute because I'm going to divide you into groups of 4. You're going to work in groups of 4 all semester. So hopefully you'll like each other. Hopefully everyone has found the paper for this week's recitation. Has anyone had trouble getting the paper? Okay, good. And hopefully everyone's found the questions for discussion. Obviously, if you're on Friday's recitation, I don't expect that you have all this stuff just yet. You don't need to answer discussion questions, just bring them. Bring your Scantrons. If you do not have Scantrons, you do not take the quiz. That's that. Okay, so we just did quiz 2. Congratulations. The average was an 8, which is really good. So people are figuring it out. I'll go over a few brief questions that seem to get a lot of people getting them wrong. A few questions I've gotten from people that might help everyone. Some people are feeling overwhelmed with the amount of reading required to take the quiz. Stop feeling overwhelmed because you don't need to read all of that. So open the quiz, and each question that you do, look up the answer. So think of it as like question about ERAD. Look up what is ERAD. You can't find it in the book, Google it, whatever you need to do. Do not read, if I have on the syllabus 2 chapters, do not read 2 full chapters. God, I'd kill myself. You can't do that, right? That's so much time, unless you want to. Don't not read it, but I'm going to guarantee that not all of that will ever be talked about in this class, so it's not effective. So to be the most effective, look up each question as you go and read about the topic. Okay, and also you should be kind of copying down these questions as you take each quiz because this serves as your study guide. Okay, so these are a few questions that people had issues with. The signal recognition particle, also known as SRP. Oh yeah, get out your letters. Get out your letters, everyone. Oh, right. So not everyone had every question because there's a pool of questions that it's pulling out. But most of the questions you'll all have are the same. The pool is like a little bit larger. Yeah, but it's worth knowing. Okay. So this talks about the function of SRP, which I have not taught you, but was really easy to look up, right? So the signal recognition particle, what does it do? A, B, C, or D? I'm seeing tons and tons of C's. Fantastic. There were a lot of strange answers, which made me think that maybe people were having technical difficulties figuring out how to select the right answer, or something's going wrong with their computer. I'm really hoping, based on some of the answers I saw. Or people just don't want to read and are just guessing. But remember, all these quizzes equal an exam. So if you get good score on all these quizzes, that's a huge cushion, right? And I'm basically saying look in your book and ask your friends. I don't do that with exams. Okay. So yes, so the answer is C. Recruits the ribosome / polypeptide complex to the ER, and we'll learn about that in gory detail. I even got some people who thought that the SRP translates the signal sequence. There's no translating of the signal sequence going on by the SRP. Absolutely not. Okay, and it says it actually in the name, right? Signal recognition particle. It recognizes the signal sequence, right? Okay. Another one. Signal sequences determine the correct localization of proteins to the. These were the choices. So, and just for kicks, I put up the table from the textbook that had the answer. Right, so I'm seeing lots of A, B, C, Ds. Yeah, so these are the signal sequences for these directing proteins to the different places. So one sequence will import proteins to the nucleus. So one sequence will actually export proteins from the nucleus. One sequence will import to the mitochondria. It's kind of like a little train ticket, right? Has the correct destination, and that's where the protein goes. If you want to send a protein to the ER, it's so easy. All you do is tag on a little signal sequence, and it'll go wherever you want it to go. Experimentally, that's really useful sometimes. Okay. So another one that people got wrong, a positive peak on a hydropathy index or plot, depending what you're talking about, represents. Great, lots of D's and L's. Good, good. And so I, just for fun, I put up a picture of a hydropathy plot. What I meant by positive is like that's your 0. Things above it are positive, things below it are negative. It kind of explains that in the book, and if you Google it, it'll come up. And so this website is really nice explanation of how what it all means. But basically, each amino acid has a score, and the more positive it is, the more hydrophobic it is. And the more negative it is, the less hydrophobic it is. So if you plot it and you've got a whole bunch of peaks, like a peak like this, a peak like this, a peak like this, those are really hydrophobic, and they're likely to be a transmembrane domain, right? Because really hydrophobic, you need a hydrophobic alpha helix that's 20 to 30 amino acids long to be a transmembrane domain. But go to the website, read about it, think about it. We'll talk about it in class. Okay. So far we've been talking about all the different ways that a protein, all the different ways to regulate whether or not you're going to get a protein and whether or not it's going to be functional. And the reason that I'm talking about this is because we're about to talk about translation, and targeting proteins to where they need to go to do what they need to do. And if you don't start with, transcription and all that, then you kind of are just starting in the middle of like just magical protein appears. So that's what we've started, and that's what we talked about. And some of you might be feeling overwhelmed with the amount of information I've given you. Some of you might think this is so easy and are waiting for me to speed up. Who's ready for me to speed up? Okay. Who is overwhelmed by the amount of information and details that seem to not be related to each other? Okay, good. Raise your hands. Good. Okay. So what I'm telling you here is this is how you can start to think about things. Start to organize your information. Because what I taught you is basically a whole bunch of points where gene expression is regulated in some way. We'll say it that way. And so what you can do is organize it. So categorize things based on the target or the outcome. For example, okay, so I made this chart, and this is an example of what you guys can do, right? So what processes affect production of a functional protein? That's basically what all the lectures so far have told you about. It's basically one question with many different answers. And so we've talked about processes involving RNA. So go through your notes and say, okay, what did she tell us about that involves RNA? How does that affect whether or not a functional protein is produced in the end? Processes involving protein, we've talked about too, right? So what processes involving RNA did we talk about, just really quickly?
Alternative splicing.
Alternative splicing, right? That affects the outcome in terms of what protein is made. What else?
RNA editing.
RNA editing, right? And changing your RNA from C to U or inserting U's. What else? So all of those are changing your sequence of your RNA, which is going to change your codons, which is going to affect your protein, right? So you list those. So there were some others, but I'll let you guys look through your notes and put them down. So processes involving the protein. So what processes involving protein might regulate whether or not you're going to get a protein, or a functional protein, I should say? Yeah. Oh, well, mRNA degradation, that would be processing involving RNA and protein in a way, right? Because it's kind of like that transition point, post post-transcriptional regulation. Yes. Right, so post-translational modifications, right? If a protein gets highly modified, it's going to, by phosphorylation, it's going to interact with different proteins. By assimilation, it's going to interact with different proteins and go different places. By ubiquitin, sometimes it'll just get degraded. And then, of course, whether or not the protein's even translated, right? If you're not translated, you're not going to have a functional protein. So I'm putting this up here really to help you guys think about how to work with the information I give you. Always look to categorize and put it into the big picture. Ask yourself, what is the big picture? What is the big question she's asking? If you don't know, ask me. Be like, well, what is the point? Because I'll tell you. That's okay. Yeah.
This is kind of off topic, but in terms of like the level of detail we'll be expected to know, I know you said you're more of a big picture kind of person. I mean, as I've been reading along in the corresponding readings, can we assume that anything that is detail focused, you'll emphasize and let us know it's important to remember this particular detail?
Yeah. Like at some point, I might say, remember glucose 6-phosphate and what it does. Yeah, if I don't mention it in lecture or on a quiz or in recitation, then it's not fair game for an exam. So if I suddenly come up with something totally weird and detailed, like draw all the structure of all the amino acids, then you have the right to come to me and say, you kind of never taught this. And so I'd like my points back. And using, of course, the appropriate mechanism for disputing exams. Okay, let's move on. Okay, so I have a practice problem. And you guys are going to hang out with your partners for a minute and try to think this through based on everything we just learned before we move on. So here I'm asking you to apply all the things we just talked about. And thinking about this and working through it is going to give you a huge amount of practice in getting used to the exam questions that I give you. Okay? So you're a scientist conducting HIV research. You notice that HIV infection results in lower levels of protein X as seen by Western blot. What could be the reason for this? And then you have to outline your thought process, so basically explain it. So for those of you who don't do Western blots on a daily basis, this is one right here. So in lane 1, it's just a marker that's just showing you protein size. A Western blot, right? So you run your gel and then you detect certain proteins with an antibody, and then you put a secondary antibody and you get a color change. So each band represents a protein. So lane 1 is just a size marker, so you can ignore it. But lanes 2 through 6, you see one band, and it's really thick and big, and it almost goes away by lane 6. Does everyone see that? So that says that protein is really abundant, probably over time, let's say. It's really abundant, and then it's all gone. So what could be the reason? So take a minute to chat with your neighbor and think about this, and think about all the things we spoke about whether or not a protein becomes a functional protein, and the kind of things you would have to test. Uh-oh.
And what else was that? Virus,
Yeah.
maybe it goes all the way back to transfer.
And be able to get.
I mean, so just put anything that's transmitted or
Well, we're trying to figure out. She's basically asking us why we believe that over.
the virus was like
Okay, guys. So, while eavesdropping, I heard a lot of really, really great words. Like words that made me think these guys, some of them at least, know how to tackle this question, which is good. And I heard some not so great words, which made me think either I didn't explain it well, which is always possible, or you guys just need a little more direction in how to tackle this problem. So let's start first of all with two clarifications that might help. One, this has nothing to do with HIV, right? Nothing to do with HIV. I'm just throwing something. You can turn it into virus X. You can turn it into condition X. I could say temperature increase. I can say anything. But basically, usually there's some outside factor that affects how the cell functions. And so that's what we're looking at. So it has nothing to do with HIV. And what we're looking at is a cellular protein. So protein in the cell is going away. And why might it be going away? Okay, so let me take some answers. Anyone feel confident? I know some people should feel confident because I heard some really great words out there. Anyone feel confident in giving me one thing that might be causing the protein to go away? Yep. Misfolding. So something's causing the protein to become misfolded? Maybe. So you're right. So I can see that your logic was that misfolded proteins get degraded, right? Okay. So you were thinking misfolded proteins get degraded. Something could be causing the proteins to become misfolded. Maybe a virus is doing something to chaperones, right? And that's causing proteins to become misfolded. Maybe. That's not the first thing I would have thought of, but it's plausible with the right argument. Other thoughts? Yes.
Being degraded by degradation.
Right. So the protein might be being degraded. Right. So that's one thing. So if you're a scientist, you would test that by preventing degradation by using a drug that stops the proteasome from working. And you could find that. You can ask that question. You can find out, is that true? Throw a proteasome inhibitor on it. If the band stops going away, then that's the answer. Right? You can look to see what happens with chaperones. And if they're going away, maybe that's part of it. Okay, give me another one.
Could the virus producing microRNA?
Yeah. Okay, so what do microRNAs do?
Inhibit translation.
Yep, they inhibit translation, and what else do they do?
They bind to the RNA, single-stranded RNA, that binds to the RNA, so it can't be translated.
Right, so they basically inhibit translation through either directly inhibiting translation or degrading the RNA. So that comes to, well, things that are before translation. So you never really had a protein, and it's not so much that the protein's being degraded, but that it's not even being made anymore. And that's completely plausible, and a lot of viruses do that. And you can look at that, and that's a really hot area of research, actually. So look for microRNAs, see if they're some kind of inhibitor of some microRNAs, and see if that protein comes back, or look to see if the RNA is being degraded. Okay, what else? Yep.
It could be modifying the DNA of the host cells.
Oh, okay. So what kind of modification might prevent a gene from ever being expressed?
Something that actually interrupts the coding of the gene itself,
Maybe.
or affects the promoter for it.
Okay, so like methylation, maybe. Yep. So might be inhibiting transcription, right? So basically, the outcome, if you prevent the promoter from being accessed, you're preventing transcription. So it might be preventing transcription. So far we've heard preventing transcription, we've heard preventing translation, we've heard affecting RNA stability, we've heard affecting proper protein folding, we've heard degradation. Those are all possible. So I actually got this question at my oral exam during my PhD. And you have to really think it through. So the way to approach a question like this is to say to yourself, okay, well, A, what's the question? Because sometimes there's distracting things in there. The question is, why is the protein going away? And then you have to think, well, what do I need to make a protein? I need transcription and I need translation. I need the protein to remain stable. What kind of things will affect whether transcription is going to happen? What kind of things are going to affect whether translation is going to happen? What kind of things might affect whether or not a protein is going to be degraded or not? And so that's how you start. You have to really take a few steps back and find out what are the basic questions being asked. Okay, you guys did a great job with that. Okay, so now we have protein. Then what? Okay, so I showed you this before. Primary, secondary, tertiary, quaternary. These are the structures of protein. But what causes these structures, right? What's responsible for protein structure, right? So basically the chemical properties of amino acids. And I feel like I kind of have to harp on this a little bit because I don't know that people appreciate it at all. People are just told amino acids make proteins, proteins fold, proteins interact with each other. But it's all based on those chemical properties. And what do I mean? So I put up here all the amino acids, and you have circled those, what are they, R groups? I don't know. The actual functional groups of the amino acids. And as you notice, they're all very different, right? So from your most basic, teeny tiny glycine all the way up to these gigantic, bulky ring structures and these sulfur-containing. I mean, they're all completely different, and the protein, the actual amino acid sequence is going to determine the structure. Okay, so what is she talking about? You guys are like, I learned this in 201, but I just want to harp on it for a minute. So here's an example. Here's your backbone, right? And you'll have these R groups sticking off, and they'll interact with each other, right? So here you have an H bond. Here you have a hydrophobic interaction. Here you have a disulfide bridge. Here you have an ionic bond, right? So that's how your secondary and your tertiary structures form, is from those amino acids kind of all interacting with each other. And then you've got protein-protein interaction. It's the same thing, right? There'll be interfaces between proteins that'll have a whole bunch of side chains sticking off, right? Right? And they're going to interact with each other. And so here it's just very basic positive, negative interactions. But mostly when you think about it, you can think of it as positive and negative interactions, right? Like charge-based interactions. Okay. Just something to kind of keep in mind. So proteins, they need to be properly folded in order to function, right? And we're going to talk a little bit about how that happens in the ER and all that. And then you'll have modifications that will affect things, and you'll have they'll determine whether or not they can bind to other subunits. So when we talk about signaling and we talk about receptors and phosphorylation and kinases and how that all functions, that's all about modifying proteins and creating binding sites for interaction with other proteins. And so proteins basically communicate the signal from outside of the cell to the nucleus to affect gene expression. If a protein is not properly folded, none of this works, right? Because all the important interfaces are not there. And so proteins don't interact with the right things, hydrophobic regions are exposed, you have aggregates, you have all sorts of bad things. So proteins fold into a conformation of the lowest energy. So what does that mean? Basically, it's kind of an automatic process, the folding. Well, chaperones come into play. But basically, their sequence is going to determine how it folds. And by lowest energy, I mean if amino acids are going to bind to each other, then they're just going to come together. You're not going to have some kind of, a negative amino acid next to a negative amino acid, because that's a really high energy to keep together, right? And you're not going to have hydrophobic regions on the outside, right? Because what's the outside of a protein exposed to? Water, right? Cytoplasm, water. So a lot of this is all about keeping the lowest energy. Pretty logical, but maybe you don't always think about things that way. Okay, so now we talk about assisting protein folding by chaperones. And basically, there's just a few different chaperones we're going to talk about. There's HSP70, and it will bind to pretty much all proteins as they come off the ribosome. That's what it's doing there. That's the ribosome. There's your mRNA, and as it comes off, it binds, and it kind of binds and unbinds to help the protein to fold. And if there's a problem with folding, then sometimes it'll get transferred to HSP60, and that's more of like a machine. It's like, you can think of it like a big folding machine, and it kind of opens and closes and spits it out and takes it back in until it's folded properly, and that requires ATP. Okay, so things to consider. Protein folding, it's, the cell's not an empty space, right? It's not just like this empty bag of water with a protein and a ribosome and some chaperones. There's a whole lot going on in there, right? It's really crowded. If you were inside a cell, it'd be really crowded because there's tons of organelles and cytoskeletal structures and vesicles and proteins and ribosomes. There's a lot going on in there, right? So it's really easy. If you're if you're a protein that's folding, you can imagine that, like, if you have exposed hydrophobic regions, it's really easy for them to start binding to each other, right? Let's say there's many proteins folding in a small space. Everything can easily get aggregated. Does that make sense? So that's pretty important to have chaperones around for that reason. And don't forget, you're having translation and folding happening at the same time. Cell's pretty warm, high temperature, so it's easy for things to not fold properly if it's too warm. And then there are degradation signals, like if proteins are not folding properly, eventually they'll be degraded. So these are the heat shock proteins. So we started talking about HSP70. It's involved in folding. Also, it's involved in translocation of proteins into the ER. HSP90 is a favorite of mine. It stabilizes steroid receptors, and it basically is involved in stabilizing proteins that are going to later interact with some kind of partner, and is currently a target for chemotherapy, but that's another story. HSP100, we're not even going to talk about it, but it's involved in unfolding and disaggregation. So when you have aggregates, HSP100 be involved. And chaperonins, which would be HSP60, and in bacteria, GROEL, GROES. And they're more of these chaperone machines that literally the protein goes in and it folds and refolds until it's right. And then there are also chaperones in the ER. And we'll talk about that in a little bit. So there's HSP70. So like I said before, binds to the peptide coming off the ribosome, and it folds, and then it unbinds. So if you have a correctly folded protein, you're good. If you don't, it'll eventually be transferred to HSP60. Here's HSP60, and so if your protein's not correctly folded, it'll bind and go into this HSP60 complex. It actually will recognize these exposed hydrophobic regions that are on proteins that are not folded properly. And it actually has a lid. Think of it as like it's an actual little folding machine. And it folds, and then once it's correctly folded, it comes out. So basically, if proteins don't fold properly, eventually they go to the proteasome and are degraded. How does this happen? So before I go on about this, I just want you to understand that ubiquitin is not only about sending misfolded proteins to the proteasome, but we're going to start with that. So ubiquitin is a 76 amino acid protein tag. You could think of it as a tag, right? And it's covalently conjugated to other proteins. So covalently added to other proteins. And it's super conserved, present in all eukaryotes, and it's conjugated to lysine residues on the protein. So if you were studying a protein and you wanted to know whether or not it was ubiquitinated and where it was ubiquitinated, you could one by one mutate all the lysines and see if you could stabilize the protein that way. So it has many functions, and one is targeting proteins for degradation to the proteasome. But there are many other functions. One is so you can have polyubiquitination, and that's what people talk about when you send proteins to the proteasome to be degraded. And that's by polyubiquitination, it means that it's a chain of ubiquitins added. So not one ubiquitin, but many ubiquitins. And so that's a very common linkages through lysine 48 on ubiquitin send proteins to the proteasome. Then there's also other ones like lysine 63, which is involved in DNA damage tolerance. Then there's monoubiquitination, so that's just one ubiquitin added, and that's involved in protein trafficking, endocytosis. If you have a receptor on your cell surface and it's signaling and you want to stop signaling, a common thing that you do is you endocytose that receptor. Makes sense, right? We'll learn about that a lot more soon. So there's a lot. I used to go to a ubiquitin conference every year. Believe it, a whole conference about ubiquitin. It was fun too. Highly recommend the ubiquitin conference. It's at Cold Spring Harbor. There's a bar on site, and they have TVs in the bar, so you can watch all the talks while drinking your beer. It's awesome. I highly recommend. Not that you should mix drinking and science, but it's actually a great way to meet people and get free stuff. Okay, so this is the ubiquitin cycle. So what do I mean by ubiquitin cycle? So ubiquitin is covalently added to proteins, and there's a process for doing that. It's not just like it randomly touches a protein and it's added, right? So there's 3 enzymes involved. There's the E1, E2, E3, very creative. Yes? Oh, you sure you don't have a question? Okay. I think it was getting lonely out there. You were like, yes. Okay. So E1 binds ubiquitin, it transfers it, it activates it through ATP-dependent mechanism, transfers ubiquitin to E2. Which one comes first, E1 or E2? Isn't it nice that they did it that way? What if it was like first E2 came? Anyways, I'm just trying to keep things lively. Okay, so E2 binds it and then transfers it to E3. But E3 is the most important one in my opinion because E3 is the enzyme that is involved in finding the specific protein to be degraded, right? There's got to be a huge amount of specificity in what protein's degraded. Otherwise, what's the point, right? If you're just going to go around degrading every protein in the cell, like, how are we even alive? right? And as we'll see later, the whole cell cycle is run on degradation of proteins. When you want to turn something off, you degrade it. So there's a ton of E3 ubiquitin ligases that are specific for proteins that are activated at certain times during certain cellular processes. And so that's the E3. And E3 grabs onto the protein, grabs onto the ubiquitin, and basically transfers the ubiquitin to the protein. And ubiquitinated proteins are recognized by the proteasome. And then there's the deconjugation event also. So as the protein gets to the proteasome, the ubiquitins are removed and they're recycled. And then the protein goes in the proteasome and is degraded down to peptides. And that's it. It really can't function very well when it's not a protein anymore, right? Questions about ubiquitin? Yes?
How detailed do you want us to know this?
I want you to know the amino acid sequence of ubiquitin. I think if you just understand there's 3 main enzymes, and the purpose is to target proteins to the proteasome. Yeah, because I'm not going to say to you what are the 3 main steps of ubiquitination. I'd never do that to you because that's like a waste of my time and your time because it's asking for memorization, right? But I might say, what would happen to a organism where the E1 was deleted? You might need to know what the E1 does, and that the whole process is not going to move forward without it. Okay, so there's the 26S proteasome, and it's made up of this center part. I'll turn off the light for a second. That looks like a barrel. That's the 20S, and it has a whole bunch of proteases. What do proteases do? Break down proteins. There's a million different proteases, and they basically cleave at different amino acid sequences. There's a whole bunch of them in there. And then there's this 19S cap, which binds to the ubiquitinated proteins and enfolds them and allows it to enter, and also has some deubiquitination activity. Why do I harp on this? It's really important. It's the whole way. Like, ubiquitination is the whole way that the cell functions, if you think about it. Because every time something needs to turn on or off, frequently something's being degraded. An inhibitor is being degraded, or an activator is being stabilized, or something like that is happening. Okay. Yes?
Is it possible for the protein to be degraded?
Yeah, thank you so much for that question. So the question was, is it possible for a correctly folded protein to be degraded? Absolutely, and that's the case most of the time. So that's why I keep saying it's not just about sending misfolded proteins to the trash. It's actually more about specifically targeted proteins that are involved in certain processes that need to be stopped. Like, for example, during mitosis. What needs to happen in mitosis? Chromosomes need to be segregated, right? You got the sister chromatids. They need to be separated so that you have cell division and one goes in each cell. So certain proteins that are involved in keeping those sister chromatids together are degraded through the ubiquitination pathway by the proteasome prior to, anaphase? Anaphase? Anyways, prior to the end of mitosis, so that it can happen. So there's no misfolded proteins at all. It's just that there's a E3 ubiquitin ligase that's specific for those proteins and needs to send them to the trash. Otherwise, you'd never separate the sister chromatids. So thank you so much for that question, because that gave me the opportunity to give an example of a perfectly folded protein that's degraded every few hours in your cells, right as you sit there. There's a ton. Yeah. Yep. It's a class. Yep. So I believe there's one E1, which is why if you have an issue with your E1, you're not viable at all. There's a handful, 5 or 6 E2s, and there's hundreds and hundreds of E3s. And then some E3s are modular, so they'll have little subunits that change. And it's because they need to be specific for the protein that's going to be degraded. So yeah, it's a class of proteins. Any other questions about ubiquitin and why I'm talking about it so much, other than the fact that I work on it? I'm telling you, ubiquitin meeting. It's actually called the ubiquitin family. And what's funny is recently sumolation and ISGylation, there's all these other post-translational modifications that are kind of taking over a little bit. You go to the meeting and it's like not as many ubiquitin talks. It's all about sumo. Okay, first stop, ER. Right, so we're going to start talking about the secretory pathway for the next 10 minutes or so. Okay, so we've got protein. It's being translated. So now what needs to happen? So we're going to start with talking about the secretory pathway, and that is the pathway that proteins take when they need to go out of the cell to the plasma membrane, to the lysosome, to the ER, to the Golgi. And I think before we start this, I want you to look at this picture here, okay? So this is the process of what's happening in the cell at any given moment with proteins, right? So you've got RNA that comes out of the nucleus, so we've talked about, right, and ribosomes bind to it, and either a signal sequence will be recognized that'll take it to the ER, and it will enter the secretory pathway, which is what we're going to talk about, and it goes to the ER, then the Golgi, then plasma membrane or secreted or lysosome. Or let's say you have a different signal sequence, right? It's actually more called a targeting sequence. That's going to target the protein. So you'll have the ribosome translate the protein in the cytosol, right? And it's going to either be a cytosolic protein or it'll be targeted elsewhere. Maybe it'll be targeted to the nucleus because it's a protein that functions in the nucleus. Maybe it will be targeted to the mitochondria because it's a protein that functions in the mitochondria. Maybe it'll go to the chloroplast if you are lucky to have a chloroplast. Or a peroxisome, or so on and so on. And I think it's important to just stop, think big picture. When I talk about secretory pathway, it's just proteins going to certain places that have to go to the ER. So not all proteins are translated into the ER. Okay? Questions about what I'm talking about? No, everyone loves it. Okay. Okay. How does a protein get into the rough ER? Well, you guys all know this already because you went through the quiz. Is this all review already? Is this all reviewed for you guys? I'm hoping that you come in here thinking it's review because you've read so much over the weekend. Okay, is it familiar? Oh, good. Okay. So here's your ER. So this is, let's turn the light off. That's better. Okay, so here's your ER. So remember, ER is made of membrane, right? And so this is the lumen of the ER, meaning the inside of the ER. It's a cross section, okay? And so here, each little dot is a ribosome sitting on the ER, on the membrane of the ER, right? And these right here, these are free ribosomes. So if it's not sitting on the membrane, so each circle here is all membrane of the ER, and then in the middle is where the protein goes and is modified and folded and all those things. And then you'll see some ribosomes just hanging out. Those are just free ribosomes. So there's your rough ER. Okay, so we'll do a little bit of experimental talking right now. So if you're studying the ER, a very useful thing is to work with microsomes. And so basically, a microsome is if you disrupt the ER, you know how lipid is and how membrane works. If you break it all up, it's going to want to bind to itself, right, because it's pretty hydrophobic. So it will form these microsomes, these kind of lipid spheres that are made out of ER membrane. Okay? Imagine you put oil and water and you kind of mix it up a little bit and it'll form all these little droplets, right? Does anyone make salad dressing? Okay, thank you. Thank you for the nods. Okay, anyways, so why am I talking about this? It's useful for thinking about how the ER works. So you'll have your ER, which is made of rough ER and smooth ER. So if you disrupt your cell, you'll get some rough microsomes that contain what? ribosomes and some smooth microsomes that don't. And so then you do density gradient centrifugation. Who's heard of that before? Okay, good. I hope everyone, because everyone did the reading this weekend. Okay, so basically, centrifugation, the heavy stuff is on the top or the bottom of the tube afterwards? Bottom. Okay, the light stuff is on the top. Okay, very good. And so does that put your rough microsomes on the top or the bottom? Bottom, right? Because it has more stuff on it than the smooth, so it's going to be heavier. Okay, so this is how you can isolate rough and smooth microsomes. Do you want smooth microsomes? Well, it depends on what you're doing. If you're just if you're studying import of proteins into the ER, do you want smooth microsomes? No, because there's no ribosomes attached. So what's the point? Okay. So something to think about when we talk about this whole pathway is about topology, as in there is the outside of the ER and the inside of the ER, and the inside of the ER is actually like the outside of the cell. Whoa. Whoa. What does that mean? Well, because the outside of the ER, when the ribosome sits and starts translating the protein in, is cytoplasm, right? And the inside of it is not cytoplasm, it's lumen of ER. And the way vesicular transport works, let's say a membrane protein's embedded into the membrane of the ER, it's going to move outward to the cell and place the protein on the membrane, so the outside should be on the outside and the inside should be on the inside, right? Because the membrane protein, let's say it's a receptor, it's going to have very clear outside and inside parts, right? No? Do you think a membrane protein is nothing and it just doesn't have any function? It's just like a decoration? It's like Christmas lights? No? Okay. Well, just remember topology, and we will revisit multiple times because it's an important concept. Okay, I guess we're kind of out of time. So we're going to start here on Wednesday. I will see everyone in recitation downstairs in a few minutes.